For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started. All right, guys, it looks like we are live, and I know you guys are excited for this one. I'm excited for this one as well. We've got David Baldock back here with us today. He's no stranger to debates. He's debated myself in the past, more of an informal discussion, though. Kent Hovind, Nephilim Free, Bill Morgan. So, David, you've been around. This is going to be a good debate. You're always cordial, respectful. So it's going to be a lot of fun. Thanks for being here, David.
Oh, thanks for having me, SFT. I mean, you're awesome, I must say. So
I appreciate the kind words. Before we, I guess, break the ice and allow you to give a brief introduction, just a couple reminders to the audience. We've got a lot of shows planned, a lot of debates and discussions scheduled. Okay, guys. So make sure you are here. Next week on Thursday, we've got Bill Morgan debating T-Jump, Creation versus Evolution. So that's going to be a lot of fun. And we've also got on the 27th, we just confirmed Nephilim Free versus Dr. Ron Garrett on whether or not Noah's Ark is real. And of course, the 24th, if I'm not mistaken, the big debate for this month, Professor David McQueen versus Jordan from Reason to Doubt on is there evidence for accelerated nuclear decay? So that's going to be a lot of fun as well. We also kind of just confirmed, we're just negotiating some details on the exact time, but first week of April, we are going to have Joseph Hubbard and Snake Was Right debating the global flood, a combination of the global flood and young earth creation. So that's going to be a lot of fun. Let me check the chat real quick. Redefine Living. He is now putting my kids through college. So thank you so much for the super sticker. Party's just getting started, guys. I'll do my best to look at the chat as I am also having a discussion with David here to save some questions, save some super chats. Let us know how the audio and video is as well, guys. We want to make sure this is a nice, smooth stream. So enough from me. You guys came here to see the debate. It's universal ancestry or independent origins. What does the evidence point to? So, David, thanks again for being here. It's been a while since you've been on. We've had a lot of new subscribers. There's a lot of new people in the audience. So why don't you just give us a brief introduction, what you're doing over at your channel, anything you'd like to let the audience know about yourself. Go ahead.
All right, thanks, man. All right, so I'm David Baldock. I recently, the most recent one I had was against Mr. Morgan on your channel. That was an awesome discussion. I really enjoyed that. I have been on sabbatical from these discussions and debates. Unfortunately, I've just been so busy over the last 6, 7, 8 months. I'm getting my MBA in April. So, I'm excited for that. And I just want to clarify, I have no science background, no science degrees of any kind. So I'm just a layman when it comes to this kind of stuff. I'm just, giving you what I know, or I'm just, telling you what I've learned and what I know and what I've heard from both the common ancestry and common design arguments. So, yeah.
Awesome. Well, great introduction there, David. Once again, to the audience, thanks for being here. Thanks for the super stickers. Why don't we get right into it then? It's going to be easy going tonight. We are going to have opening statements followed by an uninterrupted 5-minute rebuttal, and then just a cordial, organic dialogue pertaining to the points brought up in the opening statements for about 45 minutes. We'll end it with some concluding statements, some questions from the audience if we can, and we'll call it a night. This should be fun. The opening statements are going to be easygoing. We're not going to drag them on too long, but roughly 10, 12 minutes or so, and just keep it easy going. So, David, the floor is yours. Take as much time as you need for your opening statement, and we'll go from there. We'll go with the flow. So, David, the floor is yours.
All right, thanks, man. So my opening is it's pretty straightforward. Unfortunately, I wish I had some slides for me to present, but my opening is pretty straightforward. It's pretty much involving genetics and our genetic similarity with the other animals and the other life forms out there in the world. Now, regarding common design, this is a very common argument that I hear from creationists. They'll say that we share about around 98% of our DNA with chimpanzees because God used similar components to design us and the other great apes. And that's interesting. Because if common design were a thing, then, for example, wouldn't we expect manatees and whales and dolphins to be to share more similar DNA with each other? But instead we find that manatees share more similar DNA with elephants and hyraxes, whereas whales share more DNA with hippos. So yeah, if common design is a thing, then that wouldn't really make sense. And then again, DNA is used in paternity tests, and scientists use the exact same methods to determine our relationship with other organisms as they do to determine our relationship with another human being, especially like, a paternity test. So, yeah, I mean, that's pretty compelling, I must say. So, and then again, in the fossil record, specifically with, say, dogs and bears, we look in the fossil record, we find anthocyanins and hesperocyan, which are dog bears and bear dogs. Or I might have got those two mixed up, but very interesting. So again, the common design wouldn't really make sense in that case. So yeah, I mean, my opening statement is pretty straightforward. So I don't really have much else to add. So love to hear what's going on next. So. Can you hear me?
Yes. I was just typing as fast as I can for for our rebuttal.
All right.
So,
okay, cool, man. Yeah.
So let me real quick. Thanks for your opening statement there, David. And once again, thanks to the audience for being here, guys. Remove your solo screen. And I'm going to get my slideshow going. Let me see. Awesome. Let me know when you can see it, David.
Awesome.
And okay. There we go. Let me know when you can see it, my good man.
Oh,
might be a little bit. Okay.
Oh, yeah, you're good to go, man. Yeah, we can see your screen, or I can. So.
All right. Here we go. And I'll put my timer on just so I don't go above roughly 12 minutes, but I shouldn't. So, all right, let's go down to where I am starting here. Thank you, David, for the opening statement. Lots of good points, lots of good topics to discuss. I want to see where I'm going to start here. All right. Here we go.
All right, man. Sweet.
Okay, well, thanks for doing this debate, David.
Thank you,
the debate tonight is universal common ancestry or separate ancestry or independent origins, as I like to call it. What model is best supported by the evidence? I would like to point out I have written numerous books on this topic, and I will say that the evidence overwhelmingly is in favor of independent origins, and that's what we're going to talk about today. First of all, I'd like to point out that the naturalistic explanation for the origin of life has been a complete failure. When the critic rejects the design diversity hypothesis, okay, I immediately point out the fact that the naturalist requires the evolution of a simple self-reproducing cell before they can even begin discussing so-called evidence for evolution and universal common ancestry. Actually, I'm just going to stop right here for 1 second. David, no worries, my good man. Are you able to mute yourself? I can just hear a little bit of background noise while I'm going through this.
Oh, yeah. Sorry about that, man.
Oh, no, that's okay. That's okay. It's all good. It's all good. Okay. So I want to start with the origin of life when it comes to the naturalist, okay? And I highly doubt, and David can clarify in his rebuttal or discussion, I highly doubt that David here is going to disagree that there exist numerous chicken and egg problems when it comes to the origin of first life. I just want to check the chat real quick from my phone and make sure audio and everything's coming in okay. Okay. All righty. Looks good, guys. Let us know. Okay. Now, I could essentially spend my entire opening on why every hypothesis pertaining to abiogenesis fails, but I will only quickly focus on one of the many chicken and egg problems. Let us look at the highly intricate information storage system of living organisms. Even the simplest of living cells. Okay, let's take the mycoplasma, for example. This has 500 letters of DNA information. These letters, this information codes for proteins, but the problem for those that reject the design hypothesis arises when you realize you cannot decode the information in DNA unless you have highly complex decoding machinery. DNA codes for its own coding machinery. DNA has the instructions to build its own coding machinery. I'd love to know what came first, the machinery or the instructions. How do you get the machinery without the instructions? And if the instructions came first, how do you read those instructions without the machinery to read it? You need everything working together right from the start. Otherwise, life would not and could not exist in the first place. Naturalists often look to the failed RNA world hypothesis. We know that RNA is more unstable than DNA. In our cells, for example, there are about 1 million DNA breaks every day. And most of these are fixed by the amazing intricate repair systems in the cell. But if RNA is less stable than DNA, and DNA requires many amazing DNA repair systems in order to survive over time, how on earth can an RNA world be plausible? Okay, I want to get on to the number of lines of evidence, and David covered some of these in his opening statement, so this is good. What are some of the lines of evidence evolutionists look to for support of universal common ancestry, okay? They frequently look to homology, transitional forms, right, as you can see here on the slide, Tiktaalik, Archaeopteryx, for example. You could look to the synapsids. They'll also look to nested hierarchical patterns, okay? All these lines of evidence, though, are agnostic to tonight's debate, and I'll tell you why. Okay, when it comes to homology, for example, we know that human designers build in homologous patterns, and we can even see homologous patterns at the blueprint level. The earliest stages of different types of modes of transportation, sedans, SUVs, vans, for example, they all look very similar. And it's not actually until the final product that we see the most differences. The Bible tells us that we are made in the image of God. This means that there is something about us that reflects the divine. We should be able to get an idea into how God may have designed the biological world based on the way we as humans design and build things. It turns out that humans also build vehicles that seem to blend the features of multiple types of vehicles. One such vehicle that comes to mind is an amphibious assault vehicle, which happens to blend the features of both a land vehicle and a vehicle built for the sea. We even have crossover vehicles that resist classification. Okay, for example, the Dodge Journey here, the new Jeep Gladiator, which is a hybrid between a Jeep and a truck, which is pretty cool. The Ford Flex, these all resist classification. It's all characteristics of design. It's good design. Lastly, when it comes to these types of groups within groups patterns evolutionists look to as evidence for descent with modification, these nested hierarchical patterns, right? The structure of life essentially is what it is, are found far and wide in the design world. In other words, we find an echo of the overall pattern of life in the design realm. We can see hierarchical patterns at the visible level and at the blueprint level. My interlocutor tonight may as well just look to the sky being blue and the earth being round as evidence for his model and his position, if he were to look at homology, hierarchies, transitional forms, for example, because these lines of evidence are agnostic to the debate. Both models can essentially explain these lines of evidence. Both models can explain why the sky is blue, for example. The reason why the evidence best supports the independent origins model, though, is because numerous lines of evidence exist that only the separate ancestry model can explain. This model also makes the best testable predictions, which is the gold standard of science. I am going to quickly cover a few lines of evidence in my final minutes here that powerfully demonstrates independent origins and rejects universal ancestry. Okay, so first of all, and David brought up genetics, so this is good. We can touch on this to see if those historical reconstructions of the past based on evolutionary assumptions truly reflect reality. So we'll see. Okay, so we have this major unsolvable problem of mutation accumulation and genetic degeneration. Mutations are accumulating far too quickly for natural selection to filter out the damage. What type of selection can David here tonight invoke that will counterbalance the accumulating damage? We also know that the entire junk DNA paradigm, okay, not only the junk DNA paradigm, but the vestigial organ paradigm has been completely overturned. Function, function, function. That's exactly what we see. They're now looking to the genome and junk DNA as their new vestigial organ argument. We know it's all been overturned. I want to point out that the design diversity model, okay, would predict that most of the genome and most of the DNA elements found within the genome, including pseudogenes. Okay, we got paper upon paper here demonstrating function of pseudogenes, ERVs, okay, endogenous retroviral functions abound in the genomes of living organisms, ALU sequences, and other types of these variation-inducing genetic elements, okay? Jumping genes, for example, involved in regulation of the genome. These are all highly functional and active, and we predict more function as we complete more experiments and study the genome. We're only just beginning to understand the DNA language. This is exactly what we're finding, though, is function, paper upon paper. Even the so-called chromosome 2 fusion has been completely overturned. Let me get to that slide here. Orphan genes, for example, demonstrate independent origins. We can get into that, those taxonomically restricted genes. But right here, even the chromosome 2 fusion has been completely overturned, which we can discuss in the dialogue portion if David would like. Now, I want to point out too, is we also have overwhelming evidence, okay, for forward thinking in genetics. For example, we have our genes, right, which make up physical parts of our cells, while we also have what is called epigenetics, which is what controls how the genes make the physical parts of our cells, okay? Our epigenome turns genes on and off as compared to changing the actual genetic code itself. Our genes are not all turned on at the same time. This would not be good at all. So this is all about control. And these are not just non-random. These are not random changes. These are actually non-random changes, okay? Good design means we need to have some type of control, a type of control that makes sure we have the right things being turned on and the right things being turned off. This is all forward thinking, which points us back to the forward thinker. Okay, all supporting evidence of the design diversity hypothesis. Okay, I'm going to end it with a couple phylogenetic trees here, and we'll move on to the rebuttal rounds. But based on the Genesis creation account, okay, we can make some extremely specific, some very precise predictions and retrodictions. These have actually been confirmed. We see low genetic diversity in humans, which is a direct expectation if we really did originate from just 2 people thousands of years ago. We also have strong evidence for 1 mitochondrial DNA ancestor and 1 Y chromosomal ancestor of all people on the planet. Due to time constraints, though, I am going to just focus on the Y chromosome for now. This is a tree right here, a Y chromosome phylogenetic tree, okay? Dr. Robert Carter took all the Y chromosomes and made a Y chromosome family tree. This tree is incredible because what we see is exactly what we would expect if independent origins and biblical creation were true. What we see is visually is an explosion from a central point, okay? As you can see here, explosion from a central point. This is an explosion essentially from who? From Noah, the last Y chromosomal ancestor of us all. But the most important aspect of this tree, guys, I want everybody to notice this. It's the length of the branches, okay? So the length of these branches that we see. This is a reflection. Essentially, it's a reflection of DNA differences, a reflection of mutations over time. You'll notice, though, by the length of the branches, that this is an incredibly easy number of mutations to get in biblical time frame. This reflects far too few mutations if deep time evolutionary history were true. We're not looking at tens of thousands of mutations here. Okay, we are looking at just a few hundred mutations, as would be expected if the Bible's account of independent origins were true. This data did not have to be true, and yet it is. Evolutionists are constantly having to retrofit the data into their evolutionary story. They did not predict what? They did not predict low genetic diversity in humans today. They did not predict the 1Y chromosomal line, nor did they predict the 1 mitochondrial DNA line. And this is the last thing I'll say. We could go into the mitochondrial DNA phylogenetic tree. I've got about 1 minute left, so I'm just going to wrap it up here, okay? I don't want to go over time. So one last thing to consider is the fact that the length of these branches, okay, some of these branches, indicate that some people can pick up more mutations in the exact same amount of time. That means it's difficult to look at the branch lengths and conclude a definitive amount of time that has passed, essentially. Okay, it makes it difficult for evolutionists to make assumptions based on their model, especially to make assumptions as to when the chimp and human last common ancestor, when that split occurred, okay? This is why the pattern and the overall data is so important. And I've got more on the mitochondrial DNA tree, but I'm going to stop it there, okay, for sake of time. And that's just at about the 12 minutes. So, okay. Here we go. Why don't we hand it over now to David? Take as much time as you need, roughly 5 minutes, but we're not going to be crazy strict about it for an uninterrupted rebuttal. I'll do the same, and then we'll go into an open dialogue. So, David, the floor is yours.
All right, cool. Thanks, man. That was a great presentation. I guess the rebuttal would involve more questions than responses. I'm not really sure what you mean by genetic damage. And you also mentioned something about chromosome 2 being debunked. I'm not sure where you got that from. I would have to see the source for that and then read the paper and, see about what type of, or, how they debunked it, supposedly. And you also mentioned at the very beginning the origin of life. That is a separate topic from evolution or from common ancestry. And there are currently many hypotheses for that. And that's still a mystery. It's not something that really has been solved yet. But that's not to say that, scientists don't have any clue. And what else was there? I believe you mentioned something about Y-chromosomal atom, if I'm not mistaken, and feel free to correct me if I'm wrong. Again, I would have to see a source, claiming that it was around 6,000 years ago. Regarding Y-chromosomal atom, and where else? What other things did you mention? Yes, the origin of life is a separate question. Redefine living. Sorry, I just saw that in the chat. So it is. I mean, abiogenesis is a separate. I mean, it is related to evolution, but it's not evolution in of itself. So I know what you're getting at, but the origin of life is still a mystery, as we know. So, and what else did you mention? If there's anything else you covered, yeah, these are more questions about, what do you mean by genetic damage? The source for the chromosome 2 fusion being debunked, and then also the source for the Y chromosomal atom, and their methodologies and stuff like that. So, yeah, I know it's not exactly the rebuttal or response that you want, but I don't know how else to respond to that. So, but without having any sources to look at. So, so. Does
No
anybody-
problem, dude. Yeah,
All
no,
right.
I just want you to, as long as you, I just want you to make sure you have enough time to make your points,
Cool.
and feel that this is fair. So what I'll do is I'm not going to take the full 5 minutes, and then we can definitely get into the necessary questions, for example. Okay. So, what I'll do, I've made some notes based on your opening statement. I'll make this quick just to cover the couple things that you mentioned. So let me see where I could start. Okay. So genetics, I would agree. Genetics is the most direct way to determine ancestry. Okay. Fossils, bones found in the dirt, geography. Okay. These things are not inherited sperm and eggs. Genetics is, genes, traits, and genetics. Now, when it comes to your argument about whales and dolphins and nesting when it comes to these phylogenetic trees, okay? Well, I want to point out that when it comes to taxonomy, okay, you got kingdom, phylum, class, order, family, genus, species, okay? People are familiar with this, these man-made classification systems. Whales and dolphins are mammals, while sharks are fish, right? Now, in terms of function, since we are looking to nested hierarchical patterns by design and function versus by descent, then when we consider function, when we consider the anatomy, okay, the phenotype is an expression of the genotype. So when we consider genetics, physiology, morphology, okay, and so on and so forth, we would actually expect whales and other mammals to nest with other mammals when it comes to phylogenetics, okay? When it comes to genetic similarities, though, so you also pointed to genetic similarities between humans and chimpanzees, okay, and the great apes, essentially. Now, I would agree when it comes to anatomy, physiology, morphology, and genetics, humans are a lot more similar to the chimpanzee than humans would be to a dog, a bacteria, or a fish, okay? So these nested hierarchical patterns are expected by the creationists when it comes to the design hypothesis. Okay. But that's why we also got to look at the differences. We got to consider the differences when it comes to genetic similarities and these analyses done. Okay. When it comes to the humans and chimps, they use highly selective data. They also cherry pick data. For example, they ignore things like these massive gaps. They ignore size differences, copy number variations. Okay, they got a lot of assumptions going into this. I also want to point out the fact that when it comes to these genetic sequences that are similar between humans and chimpanzees, there are incredible differences when it comes to epigenetics, when it comes to genomic networks, when it comes to the gene expression, okay? So there's actually massive, massive differences, even in the similar genetic sequences, okay? Another few overwhelming lines of evidence that demonstrate humans are not related to chimps, and we can get into, has to do with multifunctional and multi-purpose genes, okay, taxonomically restricted genes, functional endogenous retroviruses. You've got unique transcription factors, just the non-coding DNA in general, okay, and all of these fascinating numbers of DNA function-related predictions coming out from the design camp. But also the FOXP2 genes, signs and lines. We can go on and on and on about the amazing genetic differences that separate humans from chimps. And when they actually consider the entire genome and not just the cherry-picked data or the highly selective data, it turns out that we are only about 80 to 88% similar to the chimpanzee, which means that's like 500 million DNA differences that separate humans and chimps. So they have to keep pushing back the split. Eventually, they're going to push back the split to the age of the dinosaurs, and that can't happen according to the evolutionary model because there is essentially a waiting time problem to actually fix all of these genetic differences. Fix means to just get stuck in place. Okay, so there's a waiting time problem for those beneficial mutations to arise and become fixed. 99% similar, okay, maybe there's a chance. 80% similar, evolution did not happen when it comes to ape to man ancestry. And I'll end it there. I mean, I can go on and on and on, but why don't I'll end it there. And I do have some slides as well that I can get into, but let's let's call it quits there, and we'll move into the dialogue. If you want to say a couple points or something to what I just said, and then we can go into some questions or something. But since I just finished talking, David, the floor is yours.
All right, thanks, SFP. I think the most obvious or the most intriguing question is, again, like the source for the 88% similarity that you mentioned, or something like that, between us and chimps. Again, I'd have to see another source, or I'd have to see a source for that, and see their methodologies, and again, why it wasn't accepted in peer-reviewed, science. Yeah, that's probably the most. That's the biggest red flag, is why it wasn't accepted in peer-reviewed literature. So, yeah.
So, yeah, if you want to start there, then good question. So even when you look to the original paper, when it comes to the similarities in genetics between humans and chimpanzees, okay, they even acknowledged the fact that there were a lot of sequences that they just did not know what to do with. A lot of gaps, a lot of copy number variation, size differences, even when it comes to the genome. So you can even see that in the original paper when it comes to the chimpanzee and human similarities. Okay. Now, when it comes to, like, for example, I showed a bunch of sources, but I know when you're going through an opening statement, it can be difficult to copy down all the sources. So let me find the sources when it comes to, so for example, here's a source that I got right on the spot. Okay. And maybe I'll get your comment here. So part of the genome is the Y chromosome, right? Obviously, that's the uniparentally inherited DNA sequence that we inherit from our fathers, okay? Then you've got the mitochondrial DNA, which is the uniparentally inherited DNA compartment that we get from our mothers, okay? So when it comes to the Y chromosome, and here's a source it's a secular source. The paper is titled "Chimpanzee and Human Y Chromosomes Are Remarkably Divergent in Structure and Gene Content." And what they discovered was that when you consider overall gene content, overall architecture, overall size differences, when it comes to the Y chromosome, the chimpanzee and human Y chromosome are less than 35% dissimilar. And they were shocked. I mean, right here is a portion from the paper. The chimpanzee MSY contains twice as many massive palindromes as the human MSY. And you can go through the paper. I got some more quotes here. More than 30% of the chimp Y chromosome lacks an alignable counterpart on the human Y chromosome. Well, here's the thing. The Y chromosome is non-recombining DNA for the most part, so it should be a little bit more stable. But yet it's less than 35% dissimilar. Okay, so here's a source that we can start with then, and I appreciate your you looking for the exact sources to what we're talking about. Take as much time as you need, but what are your thoughts on that incredible dissimilarity? If humans and chimps split from a common ancestor, say 6 to 8 million years ago, how do you explain such amazing dissimilarity between apparently our closest cousin? Go ahead.
Well, again, I got a name down, Jennifer Hughes. I don't know exactly who that is. And I'll have to look up this particular article and read it. And again, I'm not a geneticist or scientist or of any kind. I don't have a science background of any kind. I'll have to check this out, and I don't understand why it hasn't been accepted in peer-reviewed literature or peer-reviewed science, peer-reviewed science literature, for example. Once it's accepted in those, then I will believe it, but I'll also check it out nonetheless.
Well, that paper from Hughes is a secular paper.
Oh, really? That is, I don't quite know. I saw a name, but I'll actually look it up right now.
Yeah, no, I mean, it's definitely something you can look at. I'm not expecting you to look at it now, but now here's the thing. When you read through that paper, okay, obviously the authors, okay, I've got some more stuff on it here where it talks about researchers were right. So researchers were, okay, so here's one. The why is full of surprises, said David Page of Whitehead Institute of Biomedical Research in Cambridge, Massachusetts. He and his team had just found that the Y chromosomes of chimps and humans are horrendously different from each other. Why did Dr. Page use the word horrendously? Because he believes evolution. So you can go on and on. For example, here it says, but Page's team found that the chimp Y chromosome has only 2/3 as many distinct genes or gene families as the human Y chromosome, and only 47% as many protein coding elements as humans. Also, more than 30% of the chimp Y chromosome lacks an alignable counterpart on the human Y chromosome and vice versa. So and I don't want any of this to be like a gotcha moment. So we can move on. So you can obviously, read the paper at some point, give your thoughts. But I will say this, when it comes to these papers and the research being done, okay, they go in with the evolution bias. They go in with the assumption that universal common ancestry is true, the assumption that humans and chimpanzees are related and split from a common ancestor, okay, 6 to 8 million years ago. So in these papers, they come up with hypotheses as to why this would be the case. But the question is, are these hypotheses, okay, do they fit reality? Okay, because I showed a source here where it talks about how, and have a look at this, David, and then give your thoughts, and we can move on. Human Y chromosome exhibits surprisingly low levels of genetic diversity. Okay, so now we have the human Y chromosome, where every single human Y chromosome on the planet, David, is nearly identical, 99.9% identical. But we've got the chimp and human Y chromosomes that are less than 35% dissimilar. Okay, so if we have low variation, but apparently we got some rapid evolution going on in the Y chromosome, then why is every single human Y chromosome nearly identical? And why does every single human Y chromosome go back to a single Y chromosomal ancestor just a few thousand years ago? What are your thoughts on that, David? And of course, I'm going to send you the link to that exact Y chromosome paper when we're done here. But yeah, go ahead. What are your thoughts, my good man?
Thank you. So I don't really know. Again, I'm not a geneticist. If this somehow would disprove common ancestry, I don't necessarily see how it would, even if it is that different. Which again, I'll have to check out that particular paper and see what's going on before I can say either way. But I don't quite know. I can't say because I haven't really read it yet. But if it would somehow disprove common ancestry, why wouldn't the scientific community accept that?
And I would never invoke a conspiracy, but here's the thing. A lot of your secular scientists that are coming out of these secular universities, you can talk to them, ask them if they've ever even read one young Earth creation technical paper. Okay, no, they haven't. So none of these secular PhDs, for the most part, have even encountered an alternative view. So when they come up with this data that's surprising, right, when it comes to the Y chromosome, well, they're not going to question the bigger picture or the paradigm, of course, of universal common ancestry. Now, they can question the details. And this is a detail that they do their best to hypothesize. Why is this the case? I've seen all their hypotheses. I've seen what they say, okay, in terms of rescue devices, what I would say for the Y chromosome, but it doesn't work because then we should also expect some highly divergent Y chromosome sequences in human populations today. And yet we don't find that. We find incredibly low variation. But like I said, I don't want any of this to be a gotcha moment. So let's move on because your next question was the human Y or the human chromosome too. So here's a source here. Okay. This is from the Ventura paper. So once again, this isn't a young Earth creationist paper. Now, I could show you young Earth creationist papers all day, but from my experience, my interlocutors typically wave them away, unfortunately, because the data is strong. These technical papers coming out of the Young Earth creation camp are very strong and data driven. But that being the case, this is the Ventura paper. Okay, it's titled "The Evolution of African Great Ape Subtelomeric Heterochromatin and the Fusion of Human Chromosome 2." So here they point out chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be composed primarily of arrays of tandem satellite sequences. So what they're saying is this expected satellite DNA, okay, it's not actually found around human chromosomes. So if there really was a chromosome 2 fusion, okay, in the human line, what happened to these satellite sequences? They're not there. So at best, we have a chromosome 2 fusion possibly in the human line, but we've got these differences in terms of this satellite DNA, large blocks of this type of DNA that's missing. So how would you, and once again, I can say if you've never read this paper, that's fine, but it's from 2012, it's a secular paper, and they point this out, that we've got some major differences, which does pose a problem for the so-called human chromosome 2 if it really was, after the so-called split, if there was a split. So go ahead. What are your thoughts, and have you ever heard of that paper before?
No. Yeah, I just looked it up. It's from PubMed.gov, and which is also the paper on the Y chromosome being different. Yeah, this one I have not heard of. Again, I'll have to check that one out. But again, the question I would have is, why hasn't this made headline in among the scientific community? That's what throws out red flags.
Well, it's definitely made headline, as it's being discussed. And I've talked to some experts on the human chromosome 2 fusion, and they're still coming up with possible hypotheses as to what happened to this satellite DNA. Maybe it was lost. Heck, maybe it was lost in the human line. These types of hypotheses. The question is, though, do these hypotheses reflect reality? Now, here's another thing that they mentioned. So here's a good one I'll point out then, okay? That one's a little bit more difficult. It's slightly newer, but even in the original human chromosome 2 paper, David, okay, they pointed out that the so-called area, okay, where you'd find the repetitive telomere sequences, right? TTAGG, TTAGG. Yeah. They pointed out that it was incredibly degenerate. Okay, why is it so degenerate? Why are there so few repeats if there really was a fusion? And one thing I'll say is you don't gain highly functional genes by slamming together 2 chromosomes, because the data suggests, actually, and I can send you papers on this, that the so-called fusion area, it's overlapped by a highly functional and essential gene. So it actually looks like the so-called fusion site is a promoter, an active promoter for that overlapping gene that's highly functional. But yeah, so are you familiar with, it's directly in their paper, the human chromosome 2 paper, the original paper, where they point out and ask the question, why is it so degenerate? Are you familiar with that?
No. What do you mean by degenerate, though?
Degenerate means that the area where you're supposed to find these, because remember, if there was a head-to-head fusion that occurred, we would have evidence, okay, of the original telomeres. The telomeres are the ends of your chromosomes, right? They're the end caps, essentially. They protect the chromosomes from damage, okay? So where the area is, we should find a massive amount of these telomeric repeats, but they only find a few, meaning it's highly degenerate. What they're looking for there of the original telomeric repeats, they're not actually found at that location. So the question is why? And if you haven't read that paper, I mean, they have tried to say, well, maybe when the fusion event occurred, the telomeres they were already damaged and degenerate, which allowed for the fusion event to begin with. Or maybe a lot of the telomeric repeats were lost since the so-called fusion event. But my point is, yeah, of course they're going to have hypothesis after hypothesis, but does it fit reality? For example, just using critical thinking skills, would that be your thought, David? If this so-called, like, for example, according to your model, when would this fusion event have occurred, would you say?
It would have occurred after the human chimp split, but exactly when that happened, I have no idea. So, yeah.
So with that area, then, okay, being so highly degenerate, would you assume then that maybe the area itself, okay, where you'd find those telomeric repeats, maybe they were lost since that time, since the split? So obviously you got the split, say, between 6 to 8 million years ago. Mm-hmm. Then a couple million years later, you've got a human specific fusion event, right? Mm-hmm. Okay, so let's play with that then. Let's play with that, something that we can have some fun with. Why then? Mm-hmm. Let's say, even if I gave you the fusion event, David, why is that evidence for human evolution if it happened after the split? It's a human specific fusion anyways. That could easily happen, according to our model, during the flood bottleneck of 8 people.
Because the common ancestor of humans and chimpanzees had 48 chromosomes, and then after what would become chimpanzees and what would become humans, somewhere down that line, that's where the fusion occurred. Exactly when that happened, I have no idea. But
my point would be, okay, you're assuming that there was the split. You're assuming why we have the difference in chromosome pairs. Humans have 23, apes have 24. You're assuming there was a fusion event. Because if there wasn't a fusion event, then that makes ape to human, ape to man evolution highly unlikely. So you're assuming that. So when we actually look at the data, though, I'll say this. When we actually look at the data, we're missing those satellite sequences. Okay. And we're also in the areas highly degenerate. So therefore, best explanation, if there really was a fusion event, was that it occurred in the human line, which is better explained by our model because we have the bottleneck of 8 people who would have easily passed it on to everybody on the planet today. I apologize. Go ahead, David.
No, you're fine, man. I think I've seen Francis Collins and Ken Miller talk about this they talk about the human chromosome 2 fusion and how I don't remember the exact technicalities, but they basically said we can see in human chromosome number 2, we can see the leftovers of what was originally 48 chromosomes, or excuse me, 24 chromosomes. So.
Right, but that's what I'm pointing out, David, that we don't see. The area is too degenerate. There's too few so-called leftover sequences, okay? What they're saying is found there is even pointed out by the authors of the original chromosome 2 fusion paper that, listen, what's actually here, okay, is highly degenerate. Why is it highly degenerate? Well, I'll point this out. There's been more papers that have come out that those sequences anyways that you do find, okay, the little sequences you find, these telomeric like sequences, you find them all throughout the entire genome of humans, and they're not specific to fusion events, and they're functional and essential. Okay, they've had analyses and papers have come out where they suggest, wow, these so-called telomeric like repeats are functional in gene expression. So now what is there, okay, that they need to explain why it's so degenerate? Well, what is found there is found all throughout the genome. So what's so special about this so-called area of the chromosome 2 fusion? That's and the biggest thing that we didn't even really touch on is the fact that the so-called fusion event happens to be in the middle of an active and essential gene. So how do you get a highly functional gene by slamming together 2 chromosomes, David?
A highly effective gene. It doesn't necessarily have to be effective. Well, why would you consider it to be effective? I mean, we're smarter than the other great apes, but we're also much weaker physically. So
no.
I don't.
I'm saying where the so-called fusion event is found, where the so-called
Mhm,
fusion site is found, it's overlapped
mhm,
by a highly functional and essential gene to the genome of human beings. And
mhm,
they've looked at that so-called site, and what they've determined is that that site is actually a promoter for the overlapping gene. The DDX11L2 gene is what it's called. So how do you
mhm,
get a highly functional gene? By slamming together two chromosomes. Now here's the thing. Here's the last thing I'll say on chromosome 2 fusion. We can go on to something else because I know you had some more questions about some other points.
mhm,
Okay. We've
mhm,
got a few issues. The fact that the so-called site is overlapped by a highly functional gene, the fact that the site itself is highly degenerate. There's also lack of evidence for the so-called cryptic centromere, where we should find a deactivated second centromere. Okay, and we're also missing the satellite sequences. So here's the thing: the evidence seems to suggest there was no fusion event, which is a massive problem for the ape to human story. At best, if we were to just give the evolutionists the fusion event, well, even they would admit that it had to happen in the human line. Our model could better explain a fusion event because we have the flood bottleneck of 8 people, where it's much more easy to pass on the fusion to everybody. So that's my point. It seems to be like it's indirect evidence for human evolution, anyways. It's not good evidence. The evidence seems to suggest, when you look at it in its entirety, that it's not even a fusion event. So that's the last thing I'll say on the fusion. Um, yeah, what are your thoughts on that? And then we can move to another topic if you'd like.
uh, well, I'm not really familiar with this particular paper or this particular source. So again, I'd have to. I wish I could, give a proper response, but that's not what everybody's looking for. So,
Well,
um,
and to be fair,
mhm,
um, you did broad like looking at your prior debates and looking at what some of your I think more specialties are, or what your expertise is. I know you like to look to like the transitional forms. I know you
mhm,
like to look to the so-called transitions
mhm,
from our last common ancestor with the chimpanzees to now, like
mhm.
habilis, the Australopithecine. So if you want, I mean, we can go into some of those topics if you'd like, especially if those are some of your more well-studied topics. So if you had a question there, I want to hand it to you. You take it anywhere you want. If you want to go to that topic, we can, but it's totally up to you, David.
Oh, thanks, man. No, it's perfectly fine. Yeah, I agreed to this. So, yeah, um, yeah. I just again, I would have to look at the what do you call it? Yeah, those two papers. I'll have to look at them more in depth and see what's going on with them and see why they're not being accepted as by mainstream science. Um, and what was the other one? Oh yeah, the 80 to 90% similarity, genetic similarity. There's another one between us and chimpanzees.
Right.
Right. Yeah, that one I'm a little confused about. So.
Well, what I'll do, because like I said, I don't want any of these to be gotcha moments. So I'm going to send you the papers anyways, the ones about the chromosome 2 fusion, the ones about the
Okay.
genetic similarities between humans and chimps,
Okay.
the
Thanks.
differences that they're finding in the genomic networks, epigenetic differences, gene expression differences in these variation inducing genetic elements, the Y chromosome divergence paper. I'll send you that. There's even more that I can get into, where
Oh,
they've now
thank you.
and they've now discovered that the human Y and the gorilla Y are more similar in overall architecture and gene content than the human Y and the chimpanzee Y. But supposedly, when it comes to the nested hierarchy of ape to human evolution, humans and chimps are closer than the gorillas. So why is the gorilla Y more similar to the human Y, when it should be the human Y most similar to what? The chimp and the bonobo. But once again, those are more papers that I'll send you.
Okay.
And
Thanks.
I know this is kind of your cup of tea as well. So that's why I was curious if you were familiar with all of the new data coming out recently. Like this is a very recent finding here where they've discovered 3 kinds of early humans living together in South Africa, where you've got some of the Australopithecines, Paranthropus, Homo erectus. So you've got the Homo genus coexisting with the Australopithecines. Are you familiar with that? And what are your thoughts on all that? Take your time, David, and kind of take the discussion wherever you'd like to.
Well, Australopithecus coexisting with Homo erectus. I don't know if Homo erectus did. I think
it says it right here. Australopithecus, Paranthropus, and is my screen shared?
Oh yes. Oh, you're absolutely right. Oh yes. So just because one species, I mean, for example, Homo erectus could coexist with Australopithecus. Just because Australopithecus may have given rise to Homo erectus doesn't mean that as soon as Homo erectus comes on the scene that all Australopithecines go extinct. It's not necessarily how it works. Some the Australopithecines that give rise to Homo erectus, obviously those Australopithecines will be dead, but not all Australopithecines will go extinct.
And good points. Okay, it's kind of like this. So what you're saying is just because all your domestic dogs, essentially, we would agree, share a common ancestor with the wolf. They've all ultimately all come from the wolf, but we still have wolves living today. We also have pit bulls and chihuahua, right?
Right.
It doesn't necessarily mean that the parents, would have to go extinct. But here's the thing. What we're finding is actually more like the pit bull and the domestic dogs living before the wolves. You get what I'm saying? We can't live. Yes, we can coexist, of course, with our parents and our grandparents, but we can't coexist with our great grandparents, and we can't live before our parents, can we?
Wait. You said us living before the ancestor? Wait, Homo erectus living before Australopithecus? I don't know if that's. I'm not aware of any.
Well, if we've got human footprints. Right here, footprints on Greek island are 5.7 million years old. They look like humans. The collection suggests that Homo and Australopithecus, this is from Donald Johanson, coexisted as early as 3.0 million years ago. And are you familiar with, have a look at these sources if you like, are you familiar with the artificial species problem in the fossil record where, and I've got a video from Lee Berger, who he himself has pointed out that Australopithecus afarensis, right, Lucy, may actually be an artificial species. What that means is Australopithecus afarensis is the mixture of both human bones and ape bones.
Wait. So you're saying that Lucy is a fake?
Not a fake. Because what happens is oftentimes you have these bone beds that are highly fragmented. Would you agree that the fossil record, for the most part, especially the hominid fossil record, is highly fragmented, highly fragmentary?
Well, we have quite a few. Well, they have quite a few fossils from many different species.
Right. But a lot of it is fragmentary. They're isolated bones. That's what I'm trying to say. So what happens is a lot of the times you get these mixed bone beds. Like, for example, with Homo habilis, you have this massive area of all different types of fossils, all different types of the remains of all these different types of animals, including humans. I mean, I can show you a paper. It talks about how humans ate robust Australopithecines. You know what I mean? So if they're eating, and especially in conquest type situations or the environmental survival type situations, we find with the the hominids. You know, for us, this would be post-Babel dispersal events, okay? So you've got humans doing their best to survive. They're eating all types of animals. They're also eating Australopithecines, okay? Well, what happens to the bones? What happens when the humans die in those areas that they're living in? Well, the bones get mixed. You got your paleoanthropologists. They're picking up the most anomalous bones. They're picking up the most atypical bones in isolation, and they're saying, look at this. This looks transitional. Or they're putting it together with, I mean, Lee Berger, you know Lee Berger, right? He's a famous paleo. Yeah, even he himself, I got a video I can send you. He says Australopithecus afarensis may actually be an artificial species. You know, the mixture of, so it's not fake. They don't do this intentionally. It happens. Yeah. It's accidental. It's not like right here, Piltdown Man. You know, that was intentional. That was an intentional fraud. These are unintentional. For example, this one, I'll yield after this one, but I don't want you to think I'm making this stuff up. Here's an article based on a paper done assessing Australopithecus sediba, okay? Human missing link fossils may be jumble of species. Okay, so she points out that Australopithecus sediba may have parts that belong to two different species. They are a jumble of species. They've been mixed up. They point out that there are not 2 but 4 individuals in the remains of Malapa. You can read Yoel Rack and Ella Ben, who are not young Earth creationists. They're pointing out that, listen, Sediba, who the evolutionists look to as the so-called best transitional form, may actually be a jumble of species, mixed bones. So yeah, I know I said a lot there, but there's a lot of sources here. What are your thoughts on that, David?
Yeah, I'm not aware of particular. I'm not really aware of this particular, idea that Australopithecus would be like a mix of remains from different animals, and which I don't really know off the top of my head. I haven't really seen that particular source, which you said you sent, which is perfectly fine, and thank you.
Well, I've seen you use this in one of your past presentations where you said Australopithecus afarensis had feet just like humans, and you pointed to the Laetoli footprint. But here's the thing. Here's the thing. There's been controversy when they first found the Laetoli footprints. They concluded, wow, these footprints look exactly like human footprints. Well, now we know, based on all these sources here, that your Australopithecines, Paranthropus, your Homo genus, they all coexisted. They lived at the same time, okay? We even know there's massive coexistence when it comes to Neanderthalensis, Erectus, Naledi, Floresiensis, Heidelbergensis, Denisovans, all one big world of coexistence, kind of like Middle Earth in Lord of the Rings. If you're a Lord of the Rings fan, you've got Middle Earth where you've got elves and dwarves and humans and orcs all coexisting at the same time, right? So why should we then, if that footprint, okay, was not actually associated with afarensis and found miles and miles away, and the original discoverers even pointed out that these look exactly like human footprints. If they were found on the beach today, we would just say, wow, those are human footprints. Well, why are we going to conclude that they belong to afarensis? Go ahead, David.
Well, no one ever said that those were Lucy's footprints, but those footprints do belong to a hominid.
Right.
I'm sorry. Right. Not the individual Lucy, but somebody of that, or an individual from that particular species. They've also found direct fossils of. Again, I can send provide references of fossils of particular afarensis feet that have been found. So they're not. I'm sorry. Go ahead.
Well, I was going to point out first, so a lot of times they'll find these bones in isolation, okay? And they don't necessarily know for sure that they belong to the same species. And that's why I'm saying where you might find an isolated foot bone that looks more human, okay? When it comes to the paleoanthropologists, there's a couple different camps, okay? The arborealists and the terrestrialists, okay? There's also lumpers and lumpers where they lump a bunch of different variations into, they lump it together as the same species, okay? But then there's also the splitters, where any little variation, they just consider it a different species. When it comes to the arborealists and terrestrialists, you've got people who believe, Lucy primarily walked on the ground, or the Australopithecines primarily walked on the ground. You've got others that say they were primarily in the tree. So you've got some from one camp looking at very human-like bones, okay, and they'll look to what's called sexual dimorphism, where they'll say, okay, this just belongs to the much larger males, or this, is indicative of tree dwelling, while the other camp will say this is indicative of upright walking or land dwelling. So here's the thing. What they're actually doing, though, and you can find paper upon paper of this, where they are looking at isolated bones of a mixed species, and they're emphasizing the human bones and saying one thing, the other group's emphasizing the clearly ape bones and saying another thing, when in fact it's just an artificial species where many different types of bones have been mixed together erroneously. And I'm not just, this isn't some conspiracy. I mean, like I said, you got Lieberger, who is,
I see what you're saying. Yeah.
Right. So, but my point is, and comment on that too, is, but I want to take us back to the Laetoli footprint. When you actually look, so for example, the footprint was like 11 feet or 11, a size 11, roughly size 10.
Mm-hmm.
When you've got Lucy, which is about 3 or 4 feet tall. So if you actually line up like a human foot with the Laetoli footprint, especially when you compare it to a chimp, I mean, you got the big toe in line, right? Looks nothing like an ape, a non-human ape is what you would say, a non-human ape foot. So, I mean, to you, just looking, would you conclude that that is some type of non-human hominid, or, what is just baffling to you? Yeah, but what are your thoughts? I said a lot there, so go ahead.
Oh yeah. Well, it depends on what those particular what those layers would have been dated to. If it were dated to around 2 million years, it depends on how old those footprints are. That way, we can get an idea of what type of animal that those footprints came from. If it was from 2 million, hypothetically, then it could be from Australopithecus or Homo erectus. Yeah, but I can't really say without having an idea of how old those footprints are. So,
all right, why don't we do this? We said around the hour mark. It's been an hour and 11 minutes. We take just a couple minute intermission because I seen a couple super chats come through. We're going to do just a couple minute intermission. David, if you got to take a water break or a bathroom break, do so now. We'll be back in a couple minutes. I'll go save some of the questions and super chats from the chat, and we'll continue this in 2 minutes, my good man. So good discussion so far. Very cordial, respectful. I appreciate you always bringing up good topics. To the audience, shoot some questions now. Since we're kind of moderating and doing the debate at the same time, this is the way we're going to do it. And we'll see you back in 2 minutes.
Awesome.
All right, guys. I think I'm losing my voice here. I missed a super chat. I missed a super chat from earlier that I'm hoping to. Let's see. I got a couple questions, but there's a super chat that flew by a little while ago that if you guys can, whoever sent it, maybe send it again. A super chat in the side chat, if you can. Oh, there we go. Okay, so I'm going to save this one. Are you looking at the chat, David?
I just wanted to mention that the Laetoli footprints are currently dated or estimated at around 3.6 million years old, and Homo erectus, some of the earliest known specimens, are around 2 million. So, and unless there's any 3-4 million year old Homo erectus specimens that haven't been found yet, then the current, best candidate for those footprints would be Australopithecus. So I hope that answers. Oh, I'm sorry. Go ahead.
Well, I was going to say, so remember with those sources that I was showing, we've got human-looking footprints. Well, for example, like the Laetoli footprints, the footprints in Crete. We've got possible candidates for artificial species when it comes to Sediba and Afarensis. So therefore, if Homo erectus or other human-like bones are being found, in these areas for them to even become an artificial species, then obviously humans were around at the same time. If they're being found together, then obviously they were in the same areas together. Does that make sense?
Right. But if these footprints are dated around over 3 to 4 million years old, then how could Homo erectus make them if they didn't yet exist?
See, so right there, you're proving my point. This is why they're always shocked, because according to the ape to man evolution story, the story behind Australopithecus afarensis, then we've got Sediba, however you want to say it, Australopithecus Sediba, who's now been ousted out of the so-called human line. We've also got evidence that Sediba is a mixed species, going up to Habilis erectus, who's living for millions of years on this earth, and eventually you got the out of Africa scenario. So here's the thing. These findings are questioning those basic assumptions, questioning that story. Like you said, well, how can this be if, humans weren't around back then? That's based on your assumption. But for us, the coexistence makes sense. Yeah, go ahead.
Well, I mean, the radiometric dating is, it's based on multiple independent radioisotopes that all converge on the same age. I mean, it's not just like an assumption. I mean, if radiometric dating if it were bogus, then it wouldn't really make sense why they would all converge on the same, answer.
But I'm not even talking about dating methods right now. I'm saying
right.
I'm saying your own side is admitting the coexistence of Paranthropus, the Australopithecus.
Not 3 to not 3 to 4 million years ago, though. Not that far back, but around 2 million years. Yes.
But those footprints in Crete and the footprints in Laetoli, they're human footprints, and they're found millions and millions of years ago. 5 million years ago, 6 million years ago.
They're very, 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 very human-like footprints, but they're dated at around 3 to 4 million years old, and there's no known,
Right. So now you're in this circle that you can't get out of, because you can't admit that they are human footprints, because they're dated to where it would contradict the evolutionary story. So you have to say, they're human-like, but they're not actually human. You don't see the circularity in that?
Well, no, it's just because there haven't been any, as far as I know, there haven't been any members of the genus. Well, Homo erectus has not been, no specimens from that particular species have been found, older than around 2 million years old. So, that's, unless there are specimens that, just haven't been found yet.
So if you've got Australopithecus afarensis that some of your more famous paleoanthropologists are saying are an artificial species, a chimeric species of ape bones and human bones. Well, what member of the Homo genus would that be?
Wait. So, I mean, out of all the individuals, so it would have been an artificial species that is comprised of over hundreds of individuals. That seems like a really big coincidence, you know?
Well, okay. So for example, Donald Johanson himself, he reported that at one time, at least two different species from two totally different genera were represented in his bones, with some of those fossils being pointed out by even Johanson looking exactly like human fossils. So why are we not to conclude that they are? Because you're saying, oh, they don't find any Homo erectus specimens. They find fossils and that look exactly like human fossils. That's exactly what you're. Yeah, go ahead.
Well, yeah. I mean, Homo erectus was very. I mean, they were a different species than us, but you know, most creationists, you know, that I know, they say that they were just a variation of humans, which is kind of true. You know, but you know, they were very, you know, they had a larger brow ridge than us and a smaller brain. But
right. So what I'm saying, just and we can go to like dating methods in a bit if you want. But what I'm saying is, what we're seeing in these assessments of, let's say, afarensis, what we're seeing is a jumble of mixed bones, including human and ape bones. That's the more accurate interpretation rather than this incredible sexual dimorphism. You've even got
in hundreds of. I'm sorry, man, but like in hundreds of cases. Like in hundreds of cases out of all the Australopithecine, like
I'm not saying the Australopithecine is fake. There was a type known as the Australopithecines, but I'm saying a lot of your isolated bones that look more human-like, that the evolutionists want to say, hey, listen, this is your mosaic feature, for example, that represents transition. No, those are actually just human bones that have been misplaced in the Australopithecus specimen. That's what I'm saying.
That have been misplaced hundreds of times, though? Like, that just seems like, you know, like, very, very unlikely, you know? Like,
you've got hundreds and hundreds and hundreds of different fragments of bones, is what I'm pointing to. And that's why they'll pick up the most atypical, the most anomalous bone. And yeah, where do we categorize this bone? It's not like it's all coming completely together. You know what I mean? Oftentimes it is fragmentary. It is in isolation. And since we now have paper upon paper, okay, and source upon source, demonstrating such incredible coexistence among even the Australopithecines, Erectus, all your members, all your Homo variants even, right? It's easier to conclude that, yeah, some of these are artificial species because they live together at the same time anyways, okay? The Laetoli footprints, the footprints in Crete. It's the evolutionists that seem to be stuck with their basic presuppositions that, hey, listen, these can't be human footprints. These can't be human bones. When in fact, you know, from our starting point, yeah, it's an easy conclusion. They look exactly like human bones because they are human bones. Yeah, go ahead.
Oh, wait, when you say human, do you mean any member of the genus Homo, or do you mean just Homo sapiens?
So it could be. So, for example, I would say that Homo erectus is a variant, probably a degenerated variant of Homo sapiens. So yes, Homo erectus being a variant of Homo sapiens is still technically human then. Here's what I would say when we're looking, and then you can go. When we find these bones, okay, typically what we find, I think, can all be categorized in either fully ape, you would say non-human ape, fully human, okay, or degenerated ape, degenerated human, okay, or artificial species. Your Australopithecines, an ape type, okay. I've actually got a phylogeny that I can show you real quick as you respond, Actually, real quick before you respond, I want to point out, before I forget, we got a $50 super chat. So
Nice.
thank you, Alicia. $50 super chat. I appreciate it. God bless you. Thanks everybody for the support. I've seen some super chats fly by. Like I said, we are working overtime to give you full-time content as well as an upcoming creation conference. So this is one step closer. So Alicia says, there's been issues with discoveries in the intergenic regions. Real breakdown on the issue of common descent in general. So I appreciate that. And let me see here. Okay. So that being said, yeah, respond to what I said there. And I'm going to make life easy by showing you a phylogeny as well. That'll help you.
Thanks.
So go ahead, David.
No. I was just saying thanks, man.
Nope. Okay, so here we go. So this is what we would say. And I think from our last discussion, you remember, and I provided some sources showing that what we see in the fossil record oftentimes is disease, pathology, okay? We know Neanderthalensis was highly inbred, the most highly inbred people on the planet. Erectus had evidence for degeneration, okay, bone pathology, for example. Homo floresiensis, like the hobbits and the Naledi, we know have evidence for island dwarfism and reductive evolution. Okay, so what we're seeing in the fossil record when it comes to these so-called variants of humans are actually degeneration. And I have a reflection of this here. Okay, you can find this in the book Contested Bones. We've got Homo sapiens, okay? And you've got living humans, okay? But you got side branches that went extinct. Denisovans, Neanderthalensis, Erectus, Naledi, Hobbit. Let me ask you this question because I was thinking about it earlier. Yeah.
Well,
go ahead, David.
Well, the sapiens didn't pre-exist Erectus. It was the other way around.
Right, according to your model, you've got Erectus that evolves into. According to your model, you've got two out of Africas. Erectus left because remember, in the out of Africa scenario, you've got humans that leave Africa. But oh no, what do we got? We got other human variants like Neanderthalensis, Heidelbergensis, Denisovans, Erectus. So it's like, what in the world? How did they get there? Well, they've hypothesized that there was an early out of Africa event where Erectus evolved, left Africa first, right, and independently evolved into your different variants. So by the time humans actually left, when Erectus evolved into humans in Africa, humans left and they met up with, because, there's evidence for gene flow between Neanderthals and humans. We have Neanderthal genetics.
Right.
So yeah, you're looking at a progression, right? So your model would say Erectus and then humans. Okay, well, if your model's true, let me ask this then. If you've got Erectus living on, how long did Erectus live on this planet for? A million years, a couple million years?
Something like that, yeah.
Okay, so you've got Erectus living on this earth for millions of years, right? So that means we should find, especially because Erectus has incredible evidence for sophistication, intelligence, purposeful navigation. Okay, they managed fire, for example. So we should have evidence for their existence for these millions of years. We should find billions of bodies. Essentially, we should find evidence for their civilizations, where they lived. Why?
Well, not billions, but yeah, I know what you mean. Yeah.
Well, if Erectus is okay, what's Erectus generation time? 25, 30 years roughly. Let's say it's close to what ours are. If they're living on this planet for millions of years, do the math. How many dead bodies would that be of Erectus?
That's a good point. But yeah, fossilization is a very rare thing. It's very, very lucky, to find even as many that they have now. So, but yeah, I see what you're saying.
Right. So I'm trying to find in my book here that I just took off the shelf. I do the calculations actually as to how many essentially dead bodies there would be of Erectus. And okay, so right here. So let's say given the evolutionary model, okay, we can assume there were about 1 million Homo erectus living across Eurasia, Africa, essentially handling a massive distribution. Okay, this is my book, Special Creation. So let us say their average generation time is 20 years. That means about every 20 years, you have another 1 million dead bodies. And so after 1 million years, we have over 20 billion dead bodies, dead people. This is 20 billion people of which we have no evidence ever existed. Because, and I was pointing out earlier how we have strong evidence for primitive technology in Erectus, right? They would have had campsites, villages, fire. We should have all this evidence. But it turns out that if we took every single fossil that we have of Erectus, they would only fill up the trunk of a car. So where's the evidence?
I've heard that. Yeah, I've heard that so far, but I've heard that particular claim about like fossils being able only to fill up the trunk of a car or something like that. Again, fossilization is very rare, and they have to be very, very buried quickly. Otherwise, it'll be a lot of their remains will most likely be eaten by scavengers. But
remember, 20 billion dead bodies, and you also got to consider the campsites. You got to consider the evidence for their living conditions. Anyways, it makes sense with our model where you've got the Babel dispersal. Because you're right, fossilization is very rare, takes very special conditions. So that's why the billions and billions and billions of fossils, for the most part, we would say were the result of the global flood, as well as some local catastrophes immediately post-flood.
There's not enough time to get lions and tigers and cheetahs and leopards and jaguars. In only a few hundred years, though, after the flood, there's just not enough time for all that speciation that quickly. There's just no way that could have occurred that fast, though.
Well, let me ask you this question then. From your model, how do you explain the origin of all DNA differences and genetic diversity, genetic variation, essentially? How do you explain the origin of all that?
Just evolution. Through mutations. That's what causes the variation.
Right. So that's the right answer. So the evolutionist assumes that all DNA differences are the result of mutations over time. You've got natural selection acting upon random variation, the random variation being the result of mutations over time. Now, the creationist, if you're familiar with the creationist model, how do creationists explain the vast majority of DNA differences and DNA variety when it comes to the nuclear DNA, that is?
The created kinds, usually, I think. But, I'm sorry, go ahead.
Well, when it comes to like the genetics, when you're looking at these, because you're saying speciation takes too long, because according to the evolution model, you have to add the diversity through mutations, which takes a long time. And then natural selection can act upon that random variation. Natural selection and recombination, gene conversion can shuffle up those DNA differences. But my point is, if those DNA variants are pre-existing in Adam and Eve and the biblical kinds, and we're looking to this design diversity hypothesis that I suggested in the opening, then those DNA differences are already there from the beginning, right? God would have front-loaded these pre-existing DNA differences in Adam and Eve. They'd have differences within them and between them. This would apply to the created kinds. That means in one single generation, you're going to get novel chromosomal combinations just through recombination, just through gene conversion. And this isn't post hoc ad hoc, because in my opening I showed, and I can screen share again, that the evolutionists are caught off guard that even through epigenetic related changes, environmental changes, we are seeing rapid adaptation. That's shocking to the evolutionists, because they say speciation takes too long. What are your thoughts on that?
Well, it does. I mean, speciation takes, usually in very rare circumstances, but usually it takes, beyond human lifespans to, witness. You're not gonna see speciation occur within a single generation. So
I would push back on that. I would push back on that because we've discovered, David, new bird species. Okay,
right.
We've discovered rapid adaptation in real time, and
in a single generation. In a single generation.
In real time. So we've observed it today in real time. As in, listen, it doesn't take thousands and thousands of years to get.
Right. We have observed speciation. What I mean is you're not gonna see it occur in a single generation. Like you're never gonna see like I don't know,
well, you can see, well, here's the thing. You and I, David, we have pre-existing heterozygosity. We have DNA differences within ourselves. For example, my wife and I, we could theoretically have 1 million kids, and every single kid would be genetically different because we are heterozygous. So take that back to a point of creation where you've got the most heterozygosity, right? Today, I think we've only got however many DNA variants we've got in people groups on this planet. Okay. Adam and Eve would have only needed about 20 million because we've got about 10 million common today. They could have lost half at the flood, and you've still got more than enough DNA variants to account for all the people that we see today. But I want to point this out and then take your time because you brought up a good point with the speciation. Okay. So check the screen. Roughly, okay, and the last time I checked, we've got about between 11,000 and 18,000 bird species today. Okay. So you like to talk about the dinosaur to bird evolution. Well, modern birds today, birds arose from theropod-like dinosaurs. Okay. According to your model, roughly what, 300 million years ago, would you say?
300? No. Dinosaurs don't go back that far. I think it was sometime around the Jurassic or the Cretaceous, if I'm not mistaken. That's when birds appear, something like that.
Wow. So dinosaurs essentially went extinct 65 million years ago. So when did when did birds evolve, roughly?
Birds evolved before the other dinosaurs went extinct, sometime around the Jurassic or the Cretaceous. I don't remember the exact.
So between 100 and 300 million years ago, then, is what I've seen in studies.
Not 300 million years ago. I don't think dinosaurs go back that far.
Well then, David, give me a number. 150 million, 200 million, what?
150 to 65. Yeah, I mean, that's a big range, but, okay, so something like that. Yeah.
Okay, so I've seen, because now they're finding birds, true birds, even before the so-called transitional forms, okay?
Right. Yeah. Nobody ever said that Archaeopteryx was, an ancestor to modern birds.
Right. But that's my point. Is it can't be 65 million years ago because that's roughly when they went extinct. So it has to be between 100 and, well, because they keep pushing it back because, here's another full bird. Here's another true bird. So obviously birds were already around. So now we got to push it back. Well, let's just say within let's say 100 million years ago. Okay. Let's go with that. Now, if today we've got roughly 10 to 18,000 species of birds. Okay. Let's go back to the flood 4,500 years ago. Okay. We've got PhD young earth creation scientists that are making predictions on speciation rate since the flood based on this pre-existing heterozygosity model. Okay. So they'll say we should then expect between 2 and 3 new bird species. And I'm only choosing birds because this is a more difficult one. I mean, I could go with lizard species. You've only got 4,000 to 5,000 lizard species. Snakes, you've only got about 3,600. Rabbits, 29. Bats, 1,200. Okay. So let's go with birds, one of your larger species to account for. Okay. Well, we've got predictions in print that we should see roughly 2 to 3 new species a year since the flood. Okay. Well, after that prediction was made, we have Okay, evidence coming out from the Galapagos Island of new bird species evolving in real time. We can see this. And we got paper upon paper about rapid adaptive change based on the pre-existing capacity to adapt through epigenetic changes, right? Non-random changes. Okay. So that makes sense. We can account for roughly 10 to 18,000 bird species with 2 to 3 a year. You have birds evolving 100 million years ago. Okay. Why so few bird species today then? If birds supposedly go back 100 million years ago to theropod-like dinosaurs, why not hundreds of millions of species? Or a million species? If we're seeing new species in real time, how do you account for that?
Extinction. I don't know. I mean, 10,000 is quite a bit. I mean, and that's, those are just the ones that we know about. But I mean, almost, it's hard to think of a species that has, that has not gone extinct or that will not go extinct. But
you'd have to have, well, I mean, you'd have to account for some pretty massive extinction, because if we're seeing new bird species in real time, and when you actually read the paper, we're seeing these new bird species form in the same way that we predicted, shifts in heterozygosity, which is just more genetic diversity, to less heterozygosity. Okay, because that's what we would say when we look back to the creation event. We are looking back to the expansion of genetic information. You're looking back to the contraction of genetic information if you're looking back to billions of years ago, to a single cell like Ancestry. So not only are we seeing new species in real time, not only are we seeing rapid change, well, do the math, 2 to 3 new bird species a year. We're already seeing in real time on the Galapagos Island. We're actually studying. Imagine parts of the earth where we're not studying and when we're not observing speciation events. So realistically, 2 to 3 new bird species a year is not difficult. So go back 4,500 years ago, 4, 8, 12. I mean, that's easy to account for. If you want us to go back, yeah, go ahead.
Oh, I see what you're saying. So you're saying 2 to 3 new species a year, and then multiplying that by around 4,700, and then that gets you close to 6 to 10,000 years. Is that it? Or
right? So if we're seeing rapid evolution today, rapid speciation events today, and we discover these new bird species before we made the prediction of 2 to 3 new bird species a year. Okay, this is a future testable prediction that young Earth creation. Yeah, go ahead.
Wait. So you accept rapid evolution?
So
I thought you were. So what are we talking about? I'm confused.
So remember, though, David, how do creationists, for the most part, explain the origin of genetic diversity and DNA differences?
Through the created kinds and through
right. So through design diversity. So that genetic variation is already built in. We don't need millions of years like the evolutionists do to add genetic diversity through mutations. No, the DNA differences are already built in. So we say the mechanisms for speciation, okay, are shifts in heterozygosity, right? More genetic variation, to homozygosity, less genetic variation. And we can also get new chromosomal combinations, new species, simply through recombination gene conversion. Shuffle up those DNA differences can result in rapid change. And what I'm pointing out is what we see today is the type of rapid change consistent with the young Earth creation model, but not consistent with the evolution model. Because remember, you have to account for millions of years of mutation adding diversity. Right, mutations add the diversity, and then recombination can act upon the diversity. So speciation events should be far slower according to the evolutionary model. But what I'm saying is
it usually is. I mean, there are very rare. There are some times where it occurs rapidly, but usually it occurs gradually. So
Right. So my main point in all this is the critics, like you did, they'll say that, hey, listen, there's too many species to account for in the young Earth creation time frame. And I'm pointing out the fact that when you actually account for all the different species, there's only 10 to 18,000 bird species, few thousand lizard species, not that many species overall. And since now we see rapid speciation events, we are understanding the epigenome a lot more. It's actually quite easy for us to account for all the species, while the evolutionists, like you said, they have to account for why there's such few species through what? Extinction event after extinction event. But without future testable predictions, which is the gold standard of science, then that's not actually science. It's just a rescue device or an explanation. So I would challenge you to send me a paper afterwards, and I don't want to put you on the spot, but send me a paper at some point that shows me a prediction on paper that is accounting for. I want to see the numbers and the math of how many extinction events are necessary to account for, let's say, 11,000 bird species. Does that make sense?
Sure. Yeah, that's fine, man. Yeah.
Okay, so now I want you to feel like this has been good. We've had 50 or so people the entire time. I hope everybody is enjoying the conversation.
Great,
why don't we do this? Because we touched on a lot of topics. We're going on 2 hours. You're a lot of fun to talk to. I'm going to give you the floor. Unless you want to ask me some more questions, or take uninterrupted time, no time limit, as much time as you need as like a wrap up. Say anything you want to say that maybe you haven't had a chance to say, or just kind of conclude your thoughts. We'll go into some of these questions and then we'll call it a night if that works for you.
No, that sounds great, man. This was a great discussion. I really enjoyed talking with you about this kind of stuff. This is awesome, I must say. I mean, I have quite a bit to learn, about genetics. So, I'm pretty excited to, take that on too. So, and I will look up, about the extinction of birds and how we have 10,000 alive today, and how the speciation is how rapid it can be, and how extinction can account for that. So I'll see what I can do, and I'll send it to you. So.
Nope. I appreciate that. So that I'll give you the final word on that topic. Now take as much time as you need and just kind of give a concluding statement, how you think the discussion went, any final points you want to address. Just kind of wrap it all up in a neat little package, as they say. And then I'll do the same. I've got some notes. I've got some notes based on what you said in your opening statement that I'll wrap my thoughts up with. But yeah, you go ahead. Easy going, no time limit, just whatever time you need. Go ahead. I always like my guests to feel like they've had ample enough time to say what they need to say, get out what they need to get out. So go ahead, David.
No, yeah, this has been a great discussion. You're awesome to talk with and engage with. So, I'm always, I can't thank you enough for having me on your channel. Everybody's, this is, so much fun. I have quite a bit of stuff to do with genetics. I'm definitely an amateur when it comes to that. So, so.
I'm on mute and I'm trying my best to act as moderator of the chat and also debate at the same time. So
no, you're good, man.
Okay.
Yeah, you're writing in the chat. Yeah, you're writing in the chats and
right.
talking. So
I'm even in your opening statement. You'll notice I'm constantly writing things down. It's never that I'm not paying attention, but especially in discussion form, I'm constantly writing down what my interlocutor is saying. I want to try and address everything, but here's what I'll say in my closing statement. It'll take a few minutes, okay? And when it comes to your so-called transitionals, when it comes to the diversity, okay, I think even creationists themselves have been somewhat guilty of uniformitarian assumptions. What I mean is when we look to animals today, okay, when we look to vertebrates today, we have 5 basic groups. What do we got, Dave? We've got mammals, birds, reptiles, amphibians, and fish, right? So a wrong assumption would be that we should be able to easily take fossil groups and place them in these categories.
Right.
Okay?
So the categories that we have a lot that, you know, the traditional Linnaean classes that you mentioned, the 5 yeah, they don't really, they kind of break down, you know, because Linnaeus wasn't really aware of, you know, the different fossil forms that existed. And, you know, when we find an animal like Archaeopteryx, like, what does that fit in, bird or reptile? Right. You know, right?
Exactly. So you know where I'm going, and I would say that that's an erroneous assumption that we should be able to look to today, and then take the fossils, and therefore have the ability to easily place them all into categories. Because, well, I will say, obviously, we find a multitude of extinct birds, extinct reptiles, mammals, okay, etc. But we do find much more diversity in the fossil record, okay? And therefore, according to our model, we find more diversity in the pre-flood world, especially if you're starting from a state of no mutations, okay, and the largest amounts of heterozygosity, okay? Now, in the fossil record, we also find creatures that even seem to blend the features of 2 different types of animal groups, like Archaeopteryx, like the mammal-like reptiles, right, your synapsids, Tiktaalik, for example. Okay, you do got these mosaics. We got mosaics today too, like a platypus. But what we are looking at, okay, is we are looking at, when we consider the fossil record, when we also consider today, okay, extant organisms, we are looking at a much larger design picture. Because remember, the post-flood world is just a snapshot. It's just a snapshot of the overall diversity that ever existed on this planet. That's why creationists, what they're doing, David, is they are considering both continuities and discontinuities, okay? So when they're doing baraminology on these fossil groups, okay, I had a paleontologist on, PhD paleontologist, Dr. Matthew McLean, who was looking at the synapsids, okay, your mammal-like reptiles, with the evolutionists say are a transitional species, and transitional group. And he looked at a number of traits and characteristics, okay? And he discovered discontinuities within these fossil groups, okay? So in a nutshell, what you do with baraminology is you want to find a group that is discontinuous, okay? And separate it out from every other group. But within it, within the groups, there is continuity. And then that way we can approximate the created kind through fossils. And what I find fascinating, and I'll send you some papers pertaining to recent work in baraminology, is they are finding discontinuities in your so-called best transitional fossil groups, like the mammal-like reptiles, like the Dinosauria. Okay, paper upon paper, where we don't find one continuous unbroken change from one type of animal to another. We actually find continuities within groups that we as creationists predict are related, and then discontinuities outside a group. So what I'm saying is we have the fossil data and the genetic related data, right? Like I pointed out in the opening with mitochondrial DNA phylogenetics, Y chromosome phylogenetics, that is pointing us to the conclusion of independent origins. And this makes sense when this is the last thing I'll say for my closing statement, is it makes sense when we consider the fact that the fossil record reveals incredible diversity, which means the pre-flood world was far more diverse than the post-flood world. And therefore, when we look to today, we are only looking at a snapshot of the pre-flood world. And that's why the evolutionists, they can look to that which they believe is transitional, when what we are actually looking at is simply pre- and post-flood diversity. Okay? So I'll yield there. I'm looking at my notes here, and I think that's pretty well everything I want to cover. Based on what you said. But I will say this. You brought up some great topics. This has been a lot of fun. This is definitely the best kind of discussions because we're getting into the nitty gritty of the models, right? So, any final words as my guest tonight, David?
No, yeah, this has been an awesome discussion, I must say. Definitely much better than Kent's and Aaron's. So,
I think that would be the definition of what's called a dumpster fire.
I know, right? Yeah, I know, right?
So let me see if there's any last minute questions for at least for yourself. And I got that $50 super chat. So once again, I want to say thank you so much for the support. It's that kind of support why we're able to put out so much content. So let me see what I saved here. And I got to say, David, 2 hours has flown by. So thanks for keeping this fun, my good man.
No. Thank you, man. Yeah, you make this, freaking awesome. So, you're the host. So you make everything so cool. So
hey, the best kind of discussions are these ones, free flowing, organic, but at the same time, respectful, cordial. So
definitely.
Here's a question from Crimson Air.
Mm-hmm.
He doesn't specify for who, but maybe we can both touch on it. I'll let you touch on it first if you want.
Mm-hmm.
Crimson Air says, is the identical human to ape genetic mutation, which inhibits the production of vitamin C, would that be, so the gulo pseudogene he's talking about, would that be more reasonably interpreted under the common designer model or the common ancestry model? If you're familiar with that example, you can answer that question. Do you believe that the gulo pseudogene or the broken gene for producing and synthesizing vitamin C in apes and humans, do you believe that is more compatible with common ancestry? And if so, why? Is pretty much the question.
Yes. It's pretty straightforward. The common ancestry, because it's just, it's easier to derive that, characteristic from a common ancestor than to, God making humans and apes separately with the exact, same type of deficiency and so forth. But yeah, so.
So I would say that, okay, so for one, the concept of pseudogenes is being overturned in that oftentimes what they've looked to and thought was a mutation or they thought was a genetic mistake or a pseudogene turned out to be functional and important to healthy life processes in the cell, essential. Okay. And I always point it out like this, is we are just beginning to understand the DNA language. And it would be like me going over to Russia to be a proofreader. I don't speak Russian. I don't know any Russian, but I am to be a proofreader, which means I'm not going to be able to tell what's a mistake, what's not a mistake. Okay. I'd be a bad proofreader. And that's kind of like what we're doing in the DNA language when it comes to genetic sequencing, is we know so little about it. We're being shocked more and more every day as to how layered, how nested, how multidimensional the genetic information storage system of living organisms really is. So I just want to point that out, that, a lot of what they thought was pseudogenes has been overturned, and they now understand as being functional. Okay. That being said, we do have genetic mistakes since creation. Okay, we inherit roughly 100 new mutations per person per generation. The Y chromosome, the mitochondrial DNA both mutate fast. So we would expect genetic breakdown, genetic mistakes, genetic damage. Okay, so there are some truly broken genes, especially genes that natural selection doesn't really need to preserve. Okay, doesn't really need to conserve. And when it comes to synthesizing vitamin C, well, humans, okay, we can eat fruit. We can kind of get plenty of sources of vitamin C. So it makes sense that when it comes to that gene, because there's something called mutational hotspots, where there's places in the genome that are more prone to damage, more prone to mutation. So the gene for synthesizing vitamin C would be just that. The chances are it's probably a mutational hotspot where, it's broken in somewhat of the same spot with humans and the great apes. But I'm hesitating to say that because it's also, when you actually read the papers, there's a lot of cherry picked data, as in one thing is the exons. Okay. If truly this mistake was inherited millions of years ago, then they should be more degraded. And there's a few other consistencies, and for sake of time, I'm not going to go over them, but the selective data, some of the discrepancies, the fact of mutational hotspots better tells me that this is a shared mistake, but not due to shared ancestry, due to shared environmental conditions. To make it easy, this is the last thing I'll say, is if I had a Chevy Cruze and a Honda Civic and the air conditioner broke in the exact same way in the exact same spot, it doesn't mean they inherited that defect from a past skateboard ancestor. It just means that the air conditioner is susceptible to breakdown due to the same environmental conditions. In the wintertime, all types of vehicles have trouble starting. You know what I mean? That's because of a common environmental problem, which results to a shared problem in different modes of vehicles. So yeah, mutational hotspots can certainly give us broken genes, real broken genes in similar places in different genetics of unrelated organisms. So I know that was a mouthful, but that is a deep topic. You can have some last words on that if you want to, David.
No, yeah, definitely. Yeah, you mentioned about making the different types of cars and how they look similar. That's a very, very common analogy. The only thing is that cars are, those are, things that we know are designed by humans and by factories and so forth. Where organisms, they reproduce, on their own. And, we know that speciation occurs, and, over time, that can, account for these large differences that we see throughout, all over Earth. So,
well, I'll give you the last word on that one. We're now going on over 2 hours. Good question from the audience. Thanks for the super chats. Great talk, great debate, great discussion. And I want to thank you, David, for doing this.
No, thank you, man.
My pleasure. I appreciate it. Can definitely get you some more debates with other creationists as well if you enjoy this kind of thing. On this channel, we're a big believer in critical thinking, so we love getting people who may disagree to come on and let's discuss our differences. So that being said, I think we're going to shut it down here. It's been over 2 hours. It's flown by. It's been a lot of fun. Next Thursday, guys, be here for T-Jump versus Bill Morgan, and check the upcoming live stream section. I'll be putting some reminders for people. So anyways, David, some last words before we call it a night tonight.
No, this has been a great, great discussion or debate, whatever you want to call it. I, love talking with you, love, engaging with everybody. And, this has been great. I look forward to, doing this kind of thing, again, in the future. And, so, yeah.
I appreciate
and God bless everybody. So,
I agree. I agree with you. You're great to talk to. You're a lot of fun. And God bless to the chat. Thanks for sticking with us. We had a good chat, roughly over 50 for most of the time. So everybody, God bless. SFT is out.